My name's Kevin Mitchell. Welcome. I'm in the genetics department here, and I'm going to tell you about the genetics course and what you might expect to find if you actually come here to do genetics. So, genetics is central really to all of biology. Basically, any organism, any cell, everything it does comes down to the information encoded in the DNA of that cell. So, if you want to figure out how a bacterium works, you can look at the bacterial DNA and how it's expressed. If you want to figure out how human cells work, you can do exactly the same thing, or plants, or any other organism. So basically, the DNA of the organism is laid out in chromosomes. These are human ones, and the sequence of that DNA codes for different proteins. Those proteins in the cell do lots of different jobs. So that's the secret, actually. That's the whole secret of life right there. And anything that happens from, say, a bacterium senses a change in its environment, it'll turn one of those proteins on, turn a different protein off. Okay? If a human cell, say an immune cell, encounters a bacterium, it will turn genes on or off, make different proteins, and do different jobs. If a cell finds itself in a developing embryo at the head part, it'll turn on genes appropriate to make a brain. If it's in the middle part, it may turn on genes appropriate to make a heart. Okay? So essentially, that idea of the information, the control of biology, all comes down to turning genes on or off. And obviously, that means that if there are differences in those genes, if there's a mutation, say this A becomes a C instead of an A, that may change this protein. That protein may not work the same way. Now you've changed something about the organism, and that change is passed on. Okay, so that's why genetics is the basis of heredity. Is it comes down to that sequence, that information that's transmitted over time, and we can follow that. We can follow it in, say, human pedigrees, where someone has a mutation that causes a disease, and we can see it gets passed on to some people and not others, and figure out which genes are mutated. But you can follow that over much, much larger amounts of time as well, across populations, across species, even across evolutionary time, billions of years. So a little bit about the department here. The department is over 50 years old now, and our philosophy really is that we have research-led teaching, which means that all of the teaching is done by the faculty who are really doing research in those particular fields. So right at the cutting edge of what we know about any of these fields, that's where we do our work. And the point really of the education is not just to tell you what we know, or not even just to tell you how we came to know what we know, but to get you to a point where you are actually a practicing scientist and doing research, figuring out things that no one has ever seen before. We have very small classes, and so the staff-student ratio in third and fourth year is about 1 to 2, and a very collegiate sort of atmosphere. We're on sort of first-name basis with all of the students, and that is, I think, a good. There's no barriers there really, so we're all part of the same endeavor. As far as the research goes, Trinity Genetics is ranked very highly. In fact, it was ranked first in the world in the 2010 QS rankings in terms of quality of publications and impact of publications. So we've been sort of celebrating that history over the last few years. The course structure when you come into genetics is like the rest of science here. You do 2 years of basic science, so biology, chemistry, physics, mathematics. In first year and second year, you can do more biology and chemistry. And then in third year, we really, when you come into genetics, we're going to give you a foundation in all of the areas of genetics. And that's from very basic stuff from molecular biology of how cells really work, up to how they make organisms and how organisms make populations and species and so on. Again, we have a lot of small group teaching, so it's not just lecturers talking to 300 students at a time. It's much more intimate than that. And we also have a program of summer research scholarships between third and fourth year. This program has been running for over 30 years. So the top 6 students get scholarships to work in a lab in the States. This could be in Harvard, Stanford, Berkeley, where The scholarship basically pays for them to work there for the summer. It's a fantastic opportunity. I did it myself actually when I was a student here, and for my sins I'm running that program as a faculty member. The course in third year really covers many different areas. So you can think about how a cell, the basics of how cells work, and these genes are getting turned on or off when a cell is doing something. We've got people here, for example, who are working on cell death, so when a cell decides to kill itself, and what are the actual proteins involved in that process. And you might think that's not very interesting, but the reason it's interesting is because for a cell to become a cancer cell, it has to disable that program somehow. And if we can figure out how that works down at that molecular level, we can figure out ways to target tumor cells so that they will kill themselves, which is something they normally don't do. So we got people working in bacterial genetics, plant genetics, basically any organism and any problem in biology you can approach from a genetic angle, because all of them rely on proteins doing particular jobs in individual cells. I'm going to go through a few examples of the kinds of things that people do in the department in terms of research and the type of things that students would be able to get involved in themselves if they come here. But just to cover this, it includes things like my own area, which is neurogenetics and development. So how does the brain develop? What's the program that's involved in making a brain? And other areas of medical genetics, but it goes up to populations and species and looking at DNA over time to see where did populations come from, where did species come from. So our third year gives you a grounding in all those areas. What we want is that you can understand not just each of those areas, but how they relate to each other. So you can go from thinking about genes in cells to thinking about genes in species in a kind of a smooth way. In the final year, you get more advanced classes, and what that means is that you can specialize a little bit more. If you're more interested in medical genetics, you can specialize more there. If you're more interested in evolution and population stuff, you can concentrate your classes in those areas. You also do independent study in a particular area of genetics. And then this is really the key thing, the senior thesis research project, where you get into a lab like this and you really do independent research. I mean, you're supervised, obviously, but what I mean is you do original research. That is, we don't know what the answer is. So the experiments that you guys do in school, even the experiments that we do in first year and second year, they're just practice experiments. We just want you to run an experiment. We know what the answer is going to be. It's not research. This is when you get to do real research, when you learn how to be a scientist and can really, discover something that no one has ever known before. So that's the thrill of being a scientist. And so let me give you a few examples of the kinds of research that's going on here. It's a few different stories. And the reason is this is the kind of thing that you could be involved in as a student. Basically, you would, in fourth year, end up in one of these labs or a similar one. So I'll start with this work of Professor Jane Farrer, who and her colleagues, who've been working on this inherited eye disorder for 25 years now. So this is a condition called retinitis pigmentosa, in which the cells in the retina here, the photoreceptors that detect light, degenerate over time. What happens is people's vision is fairly normal up until they're about 40 years old, and then these cells start to die, and they start to die mainly from the outside in. And what happens is your vision, which would ordinarily be like this, starts to go like this, starts to narrow down, and eventually these people go blind completely. So it's a devastating condition. It runs in families, and about 25 years ago, they were able to find a very large family in Ireland where this condition was being inherited. So all the people who are colored in black here have the disease in this enormous pedigree. So what they could do is say, actually, somewhere back here, in this person, a mutation must have happened, okay, that has been passed on to these people, but not these people, or these people, but not these people. And by figuring out which bits of the chromosome were passed on to those people, they could narrow down the region where that mutation must lie. So the idea is somewhere in the DNA, there was a mutation that changed some protein that causes this disease. And it turns out the protein is made in those photoreceptors, and it's responsible for detecting light. And when it's mutated, it causes those cells to die because it makes it toxic protein that actually gets gummed up in the cells and kills them. So the problem here is not that the protein's not working, it's that it actually becomes a toxic protein. So they went from that to that understanding of really basic science to generating a model where they could do some experiments and see if they could fix that. So the way that they did that was make an animal model. They can take a mouse, mice have the same genes that we have, mutate that gene in a mouse in the same way, and those mice go blind, and their retinas degenerate, and then they could figure out a way to try and fix that. And the way that they do that is to use this molecular technique to target that gene and turn it off. Okay, so it stops making the ordinary protein. And that has been extremely successful in the animals, a very exciting stage to get to the research to that point. And now they're developing that technique through a company that's been set up here to actually get that to a stage where they can do clinical trials in humans with a gene therapy to correct that genetic defect in those people before they go blind. So it's a nice story because it goes from very basic science to this potential clinical application, but it also is a lesson in how long that takes, because this has been a 25, 30-year kind of effort. Okay, let me tell you something completely different. This was a story that also starts with really, really basic science and ended up somewhere very unexpected. So many years ago, people here were working on cattle. So they were interested in the question, a historical question, where were cattle domesticated? And were they domesticated once or twice? So who first had the idea to take a wild buffalo and bring it into a campfire or a village and actually make domestic cattle out of it? And so they could use DNA tracing to look at the relationships between cattle species in India and Africa and so on. And what they actually found was that cattle were domesticated twice, separately, once in Africa, once in India. And then you could look at the migrations of those and where they went based on the degree of genetic relatedness between cattle that are found here and cattle that are found there. So that was, again, very basic research, just an interesting academic kind of a question. But it led to an idea that if they could trace cattle across this huge distance and across tens of thousands of years, they could certainly trace your steak back to the farm that it came from. Okay? And that became interesting when the BSE scare and other diseases were around that could be passed to humans, because then it becomes important to know if your steak came from a farm that has BSE or doesn't have BSE. And so they set up a company called Identigen, which has been very successful. And if you see this DNA traceback on your steaks in the supermarket, that's this company that came out of the department here. So a very successful commercial entity now, biotech company. And these guys were also the ones who found that the so-called beef in the Tesco burgers there, I think it was Tesco, a while ago, was actually horse meat. Now you can do the same thing, that tracing populations for human populations as well, which is a really interesting area these days. And one of the things that Professor Dan Bradley is doing here is trying to trace the Irish population. So you can see there were people here prior to the Celts, then the Celts came in, then Vikings came in, then other people came in and so on. And you can look at the signature of those people that you can still find in people today. And you can see different concentrations of, say, Viking Y chromosomes in different parts of the country and so on. And actually what they're doing now is really interesting in that they're going back to the remains of pre-Celtic era peoples who, for example, would be buried beneath something like this, a dolmen. Okay, and so here's one of his students who's actually taking DNA out of about a 5,000-year-old skull that they can now analyze, because that DNA is incredibly well preserved, and they can figure out something about what these people were like, who they were related to, even possibly things like what diseases they might have died from. Okay, so those are a couple areas. We've got people working on many different sorts of aspects. This is just an example. One of the people is working on the genetics of flower development. So what is it about the genes of this plant that makes it make a flower with this pattern versus a flower with that pattern? Now, these are just nice, pretty pictures of flowers. You might think that's not important. But of course, if a plant doesn't flower, then it doesn't breed. And so this kind of understanding this basic biology is really central if we want to figure out and address the problems of feeding our growing world population, which is going to take more than the basic kind of farming methods that we have at the moment. I mentioned the cell death and cancer. This is Professor Seamus Martin, who's really one of the world's leaders in this field of understanding what it is, what are the biochemical pathways within a cell that tell it to kill itself. And when a cell becomes cancerous, how does it actually switch those off, and how can you switch them back on and prevent cancer? And he's made some amazing progress in that area over recent years. Let me just finish by telling you a quick story in my own area, which is neurogenetics. So this is trying to understand how the brain gets put together, how the brain functions, using genetics as a tool to do that. And this is a picture of a mouse embryo showing the nervous system, where this one particular gene is made in the nervous system, but it's not made in other parts of the embryo. And we can see the pathways in a mouse brain that connect individual parts of the brain, and we can go down to the level of individual neurons as they're growing, trying to find a partner and figure out who they should connect to. And then we can bring that back to the level of human brains. This is actually my own brain, which is awesome. It's actually very completely ordinary, disappointingly. And so we can figure out, is there, when we learn something in mice, can we relate that to humans? And the reason that we're interested in that is that the information to make an organism and the information to make the brain of that organism is encoded here in the DNA of the fertilized egg. So as that embryo grows and cells start being differentiated into brain cells or liver cells or skin cells and different parts of the brain, those all involve this genetic program of development, and we've been trying to figure out what that is. And one of the key things in brain development is that those cells have to connect to each other. So they make these incredibly complex and beautiful projections of nerve fibers that connect different parts of the brain to each other. And if you look down at the level of really individual cells, this is a little cartoon of just one little part of the brain showing a few of the different types of cells. So these are all different types of cells in the brain that have different jobs to do, and they make connections to each other in really, really specific ways. So what we're interested in is finding out at the really molecular level, when two cells touch each other, how they know if they should connect to each other or not. So what this is a picture of one nerve cell touching another nerve cell, and these are some of the proteins that stick across the membranes that let them recognize each other. The reason this is interesting is because if you get mutations in those proteins, then the brain develops in the wrong way, and that can lead to conditions like autism and Fragile X syndrome and epilepsy and schizophrenia. So trying to figure out these things and model them, trying to find mutations in humans that lead to those conditions, and trying to understand the basic biology is what we do. And recently we found a mouse that has a mutation in one of these kinds of genes that ends up with epilepsy, and also a kind of a hyperactivity that's similar to what's seen in ADHD, in that it responds to stimulants by becoming less active, which is true for ADHD patients, but not true for other people. And actually, this is an area that's been really taken off in the last few years. A few years ago, we understood very little about what caused autism or schizophrenia. Now we can account for about 30% of cases of autism, at least, by finding a particular causal gene in those patients, and that's growing all the time. So that generally is an area, this human genetics, that's absolutely exploding right now and will continue to explode and be a huge growth area in science and also in all of the industry and clinical areas that come out of that. So Angelina Jolie is up here recently, because you probably heard. Breast cancer runs in her family. Her mother died of it. I think she has 2 aunts who died of it. And so there's a mutation there that's being passed through her family, and it's a mutation in a known gene called BRCA1. And she was tested recently to see if she carries that mutation, and she does, which means that she had about an 80% risk of developing breast cancer by the time she's 50. And to avoid that risk, she made the decision to have all of her breast tissue removed, which is unfortunately the only thing that can be done at this stage. But it did mean that genetic information probably saved her life. And it reduces her risk down to about 2 or 3%. So that's an illustration of the the power and the importance of that genetic information. And what's going to change medicine now is the availability of that information, because it's now incredibly easy and routine to sequence an entire human genome, to not just do test one gene, to get all of the genetic information of an entire genome. So this is the cost of genome sequencing from about 10 years ago. The first human genome cost about $100 million to sequence, and it took 10 years and hundreds of people working with massive factory-scale sequencing machines. This cost of sequencing has gone down to where now it's about $1,000, and it can be done in a day. Okay? So it's going to get to the stage where when you go to your doctor with some complaint that they can't diagnose, it'll be cheaper for them to just get your genome sequenced than for them to spend any of their valuable time thinking about whether or not to get your genome sequenced. So what that means is there's going to be a huge demand for people who understand this stuff, who can interpret genetic information, who can tell a doctor, if that test gets done, what that information actually means. And that's just going to continue. This is just an illustration of the change. So these are the old DNA sequencing technologies from even 5 or 10 years ago, these massive warehouses of sequencing machines. This is the new one. Little thing that actually sits in the palm of your hand and can be plugged on a USB stick right into your laptop, and you put some blood in there and it reads the sequence out in about 2 hours. So that's a game changer, and it's going to change not just genetics, but genetics is going to change medicine and many other areas of science as well. Okay, one brief note then on our alumni, and many of them, about 2/3 go on to do a higher degree, so a master's or a PhD. We've got lots of people doing who go into academia and become professors and all over the world, really, in top universities. There's obviously a lot of growth in biotechnology. I told you about 2 companies that have come out of genetics here at Trinity, and the one that traces meat and the one that's developing techniques for inherited eye disorders. And then people have gone into medicine, law, journalism, funding agencies and regulatory agencies and so on. So there's a broad, whole variety of areas that you can go into with a degree like this, where you're either using your scientific degree or at least you're using the scientific education in critical thinking that is very in demand among employers.